Hi, my name is Amy Pierce. I am a senior biosystems engineering major here at the University of Arizona, and I am currently interning in Dr. Barry Pryor's mycology laboratory. Mycology is the study of fungus, and my project is focused on creating a field test to help farmers determine if their crops are infected by the lettuce wilt pathogen Fusarium oxysporum lactucae, or FOL. At any given time, there are an average of 4 people working on this project. When I started, I was working under a postdoc with 2 fellow undergrads, but now I am the project coordinator, responsible for coordinating the activities of my fellow interns. I am responsible for keeping a lab notebook detailing what I do each day, any calculations I make, results of tests, interpretations of these results, as well as an explanation of why my results may differ from what I expected to get. The main test I conduct is PCR, which I use to amplify a region of DNA in the FOL pathogen. Along with conducting these tests, I conduct PCR primers, which determines the sequence of DNA to amplify. First, we need to design and test primers that will amplify the Fusarium pathogen, but won't amplify any other DNA sequences. For example, Fusarium oxysporum opuntiarum is not what we're looking for, but has only a couple of nucleotides that are different from FOL, so we need to make sure we choose a primer that includes those differentiated nucleotides. Once we are sure that these primers are effective and consistent on the fungus strains from our Petri dishes, we will test these same primers on lettuce roots infected with the pathogen. Once the DNA is amplified, I conduct gel electrophoresis to visualize the Fusarium. And this is an image of the gel electrophoresis setup. So, to do this, we use an agarose gel and submerge it in a liquid that has the same concentration of buffer as the gel. Next, we inject our amplified DNA into the holes of this gel and run a positive voltage across it. DNA is negatively charged, so it is pulled toward the positive voltage. To see if our DNA was in fact amplified, we image the gel under a UV light. Along with conducting these tests, I also maintain the fungal cultures, so we always have happy, healthy pathogen strains ready to sample. So, I really like that this is a hands-on experience. I'm not just learning about lab equipment. I'm getting to use the lab equipment. I also like that this internship requires critical thinking. When a machine isn't working correctly, or my gel images aren't giving me the results I want, I have to figure out what's wrong and fix it. It's also important to me that I'm doing meaningful work. I don't just wash dishes and clean the lab, although I do those things. I'm actually contributing to a research project. I don't like that there wasn't a lot of training involved, and lab protocols are hard to find. I don't like that information doesn't seem to get communicated to the entire lab, so often people are on different pages. I also don't like that getting results in a research project can be a really slow process, but these dislikes are pretty common in almost every lab I've been in, and I've been in about 4 laboratories now. I found out about this internship by watching videos on the University of Arizona's Controlled Environment Agricultural Center's YouTube page. I saw one video of Dr. Pryor presentations, and I loved that he had so much energy and enthusiasm for fungus, so I sent him my resume and asked if he could use help in his lab. So I guess I didn't necessarily find out about an internship so much as ask Dr. Pryor if he could create one for me. So, would I recommend this internship? Absolutely. If you are ambitious, want to learn, and don't need someone hovering over your shoulder to get your work done, this is a fantastic lab to be in. Dr. Pryor is a person who loves to see students learn, and if you have a project that you want to work on, even if he's maybe not working on it at that time, he will encourage you to research it. He'll give you a space to do it. He might not have the funding to do a project that is outside of what he's already doing, but he'll give you whatever resources he can. So if you're interested in fungus at all, I think you should contact him. Thank you for listening.